All right, let's move along into the past now, into deep time, and try to get a handle on the fossil record, starting with the origin of living things and the origin of ecosystems. We have a basis in microevolution with which to think about these data, but we'll also be talking about macroevolution at these new scales. So we'll start with an analysis of the fossil record. What is a fossil, we'll ask, and describe how fossils are formed, and then hit some of the major hallmarks in the evolutionary process, some of the major events that you can use to help structure your thinking about deep time. There are a few dates along the way that you can just commit to memory. They're handy to create a sense of scale when thinking about the deep time record, because it's hard for humans to grasp these great spans of time. It's not natural for us. We who think on the scale of generations, maybe, parents, grandparents, great-grandparents, not distant ancestors like the ones we'll be looking at here. So imagine yourself walking across a landscape, dry and crumbling like this one in South Africa, and you can do that in a dry area in South Africa, and see something like this on the ground, a little mound of stuff on the ground, and you recognize that it's something you're interested in, so you put down your box of matches for scale there. There's your rock hammer, as every geologist has a rock hammer, and you clink it open with your hammer, and what do you find inside? This little creature, all curled up inside that fossilized burrow. So this animal curled up here in the burrow was trapped in that burrow by some mechanism that preserved it over hundreds of millions of years in the condition in which it died. And numerous creatures like this have been found in a similar position in burrows like that, and the suspicion is that they died while estivating, which is like hibernating, but you do it in the summertime when it's hot and dry. So probably these little creatures were curled up, preserving their energy, holding in their water in a state of torpor, and got caught in their burrows. And then the paleontologist has the whole skeleton, a nice preservation of an entire skeleton to help reconstruct this organism and its relatives. And this is a relative of the ancient lineage to which we belong. More of these burrows have been found, and here's one that was scanned, and so that the internal anatomy of what was happening in the burrow could be seen via a scan. And not only is there that little therapsid in the burrow, just like the one we just saw, but there's a little amphibian in there shacked up with it. It has a few broken bones that had healed in its ribs, so it was an animal that had lived a long life and healed some breaks, somehow climbed into that burrow with the estivating therapsid, and were preserved together. In an artist's reconstruction, you can imagine it happening thusly. Amphibian walking, entering a burrow with estivating therapsid. You can start to tell stories about natural history based on fossils like these. You're limited only by your imaginations. In a scientific context, you'd better frame your imaginative stories according to some standards by which the stories may be tested or further investigated. Otherwise, you're just telling stories. And that's a criticism that's been leveled at paleontologists, archaeologists, and many others. We had a request just to be careful with these doors in coming and going, because they're so loud when they slam. So just try to remember to close them, and close them slowly. So paleontology, paleoology, ontology. Ontology is the study of being, and paleo refers to the ancient past, right? So paleontology, and there's a definition of paleontology if you want. A study of prehistoric life, simply. Prehistoric. Historic referring to the portion of human history that's documented usually by writing or maybe artwork some form of cultural registration of information, the prehistoric record, or the study of biological history across deep time, all levels of organization. It's a very broad field, and the best approaches, contemporary approaches, are paleobiological. So if you speak of a paleobiologist, that's someone who's taking this broad approach and studying living organisms to assist in the reconstruction of ancient organisms. It's not just a dusty old science of studying dusty old fossils. It often incorporates the study of living creatures in the lab via computer simulation. It's a rich field. And the campus here, the departments here excel at this. And it's one of those places where Berkeley stands out for its work in paleontology and paleobiology. And that's been true for a very long time. A lot of courses here in this subject. A huge museum full of fossils downstairs. Fossils. The remains are traces of past organisms. The remains are traces. So let's look more closely at different types of fossil preservation. Here's a big skeleton. If you can see it, there's the head. There's the back of the body. A big, almost rhinoceros-sized, big skeleton. Now, those are a registration of the ancient bones of that animal. The bones may have some of their original organic content, or they may be mostly replaced by materials from the environment that entered the bones and replaced the contents of the bones. We can speak of intact fossils. An intact fossil, well, this is clearly an intact fossil. This is a mammoth that's thawed out of ice, where some hikers find this creature on a receding glacier or something like that. It doesn't happen altogether infrequently that people find mammoths melting out of ice in the far north, and those are the original tissues. You can make a mammoth burger if you want, and people have, out of something like that. A woolly rhinoceros was just found a couple of weeks ago. For the first time ever, a woolly rhinoceros, an ancient type of rhinoceros, was found melting out of ice. First one of those. So these are intact fossils, or insects in amber. That's the insect in the arthropod there, in the amber. Its whole body intact. And perhaps DNA preserved, although the molecular information in this organism does decay over time. So there is an opportunity to get DNA out of here. You've got the morphology nicely. But an opportunity to get the DNA exists if it's sufficiently intact. It tends to fragment. It decays. Those proteins decay. But even fragmented with contemporary techniques that have just become online in the last few years, you can work with very fragmentary DNA and start to place an organism like this, potentially, into a cladistic analysis with molecular characters. Now, heating and cooling of the amber in general might denature that molecular information to too great a degree. Another type of fossil preservation. Here, the original tissues of the organism also might be present, but it's two-dimensional. It's flattened, right? It's compressed. It's a compression fossil. In the form of petrified wood, we have the replacement of the cavities in the tissues of that plant and even the atoms, the original atoms of the organisms themselves, atom by atom, with exogenous environmental materials, creating a very faithful reconstruction of the organism, but not the original tissues of the organism itself. Maybe none of the original organic material preserved in a case like that. Molds and casts. If you think about what a mold is, you can make a mold of your teeth at the dentist's office. That's an impression of your teeth. It's a negative registration of your teeth. Or you can make a cast from that mold, which is a positive reconstruction of your teeth. It essentially looks just like your teeth. The cast does. So molds and casts can form in the fossil record based on, for example, shells that fill with sediment. If you have an empty shell and it fills with sediment and then the shell dissolves away, you have a cast of the internal structure of that organism. Or if the shell falls on mud and then the shell is removed but the impression is left, that could be a mold of the specimen, a negative of that original positive of the specimen. Trace fossils in general refer to the impressions, the signs that an organism leaves during its lifetime that themselves could be preserved. The organism is long gone here. It walked across this landscape, leaving footprints. But the organism is gone and didn't get preserved. But the traces of it did. It's footprints. Footprints are actually very helpful in reconstructing a lot of aspects of biology. But the organism itself is gone, just its trace. Another example of a trace fossil would be a coprolite, just for fun, you don't need the term, but the feces, the fossilized feces, holds abundant information about organisms. For example, their diets. So those would be trace fossils as well. Taphonomy is the study of how fossils form. That's important, and usually as part of a major paleontological expedition, you'll have taphonomists available to assess that fossilization process, the sequence that led to the development of the organisms that you're analyzing. Imagine these little guys caught into these hollows in this plant. If the stems of these plants are hollow and the organisms are walking along and occasionally fall in, they'll get trapped because of the scale of this plant for this organism is vast. And it's caught in there and it can't climb out. And if the waters fill over with sediment, it may trap these organisms if they've starved already, or just trap them in there and bury them. And this is a real-life case where paleontologists find these little guys as little collections of skeletons in these ancient habitats where they were caught. The taphonomy would be an analysis of that process. Shallow water environments are very typical media for the preservation of fossils. Organic material falls in, and in these shallow water conditions, and if they don't decay rapidly, the organic materials, they may be covered over by silt and sediment slowly and preserved. So the interface between water and land is a great preservational environment if decay doesn't happen too rapidly and if the waters are not too rough. In these shallow water lake environments, places like that, excellent fossilization media, and very important for the human fossil record. Much of our human fossil record from Africa comes from environments like this. We'll see that. We'll have a lecture and a half or so just on human evolution toward the end. Up here in the hills of Berkeley, you get these giant stick nests. There's at least one group in the field section of this course studying wood rats with these big stick nests. They stand tall, these nests. They can be this high, they can be shoulder high, higher than my head. They build up over the generations. The female wood rats build them up and pass them along to their daughters in a matrilineal heirlooming process. So they build up over the generations and are maintained. At least that's how the story goes. And they're prominent features on the landscape. These wood rats are very closely related to the familiar pack rats. If you're from the desert southwest of the United States, you've heard of pack rats. They live in very closely related, same genus. The Neotoma genus is our local guy and is the guy that forms these middens in cave environments. These pack rats also accumulate stuff. They just like to collect stuff. They neurotically just collect stuff, including anything that might be out on the landscape, bones and feces of organisms and leaves and sticks. And they bring it all back into their nest, and then they pee all over it. And then in peeing on it, their pee is so much lacking in water in these environments that when it crystallizes, it forms this hard crystalline coat and crystallizes all this stuff together, and it will be preserved indefinitely as long as the cave environment stays cool and undisturbed. And these are a major source of fossil accumulations for paleontologists in the desert Southwest, important in the reconstruction of ancient climates, the reconstruction of ancient ecosystems. So you actually have an organism that's collecting the stuff and it's getting preserved, pack rats or wood rats. Kind of a gruesome experiment here with a lizard that's died and subjected to decomposition via ants. And you can see that first the soft tissues of the organism are removed, and pretty quickly you get down to the bones. And what's going to happen to this organism if it's just out on the landscape? Forget the wind and water and the trampling of the deer. Just the insects alone, the little ants themselves, are dismembering this thing. And over time, you're losing all that information. You've lost the bits of the tail. They've been separated. The limbs are almost off. The pelvis is still on there. Pelvis is still intact. The skull's there, fortunately, because that's always really important with information. But you've lost so much just via invertebrates. You're losing your skull now. And you're also dissociating everything. So the likelihood of finding a whole skeleton is actually relatively rare in paleontology. It's much more frequent to find single elements. Imagine a much larger creature like this having died. A couple possibilities. It may get buried in toto, the whole creature, sedimented over. A big rising of the river, all this sediment, imagine, might flood this creature over and bury it intact, the whole thing. Or it gets ravaged by scavengers and wind and storms, and it breaks apart such that only a handful of bones are preserved. Two avenues of possibility shown here. Well, in the one case, it's intact in the soil and is maintained. Remarkably unlikely, but possible. When the soil starts to erode away, maybe you lose the whole tail. But by the time it gets to the base of the tail, your paleontologist arrives to find it and is able to pull out the rest of that skeleton. That would still be a remarkable find, remarkably intact, having just lost a bit. More likely, it will have been broken up, dismembered, and maybe one element of it preserved, maybe just the humerus or just the femur. And so the paleontologist at least gets one big bone out of it, but that's all. Having assembled a collection of fossils, one of the critical aspects to help interpret them is how old they are. And this used to be a rough science of dating fossils. Remember Steno, the all-important Steno, and his insights about the law of superposition, right? That sediments further down in a column are older than sediments higher in the stratigraphic column. With that insight, geologists like William Smith were able to identify certain fossils that were characteristic of particular strata in the column. And having identified these index fossils that could be correlated with particular strata, for example, the yellow beds, because some soils are yellow, some are more pink, some are more red. Well, if you can identify the index fossils of the yellow beds and the index fossils of the red beds, and you know the red beds are higher, then you can say that these fossils, based on their morphology, are older than these other fossils that are higher, because they may not overlap at all. They may have changed morphology over the sequence of strata, and they do. So by identifying index fossils and mapping the geological conditions of an area like Great Britain, William Smith was able to estimate the ages of these sediments based on their index fossils. He didn't know how old exactly they were. He didn't know if they were 20 million or 60 million. He just knew, well, this one's older than this one. And that alone was helpful. They could make estimates of age based on other assumptions like erosion rates, and they did, but they were very rough. But they at least knew older from younger, and they gave names to those different time groupings. This was a relative dating system. After World War II, advances allowed for absolute dating systems, where actual quantitative estimates of age could be made based on radiometric or radioisotopic dating techniques. Radioisotopic dating techniques. Remember that there are isotopes of atoms? You have your periodic table here. All these atoms, an atom is defined based on the number of protons in its nucleus, right? So each of these different atoms has a different number of protons in the nucleus. But each of these atoms can vary in the number of neutrons that they have in their nucleus. And the different numbers of neutrons for each atom is a reference to these different nuclides or isotopes of each atom. Some isotopes, some elemental isotopes, are unstable. They decay over time. They radioactively decay to another form, to another nuclide. And those rates of decay are measurable. Different nuclides have different half-lives. We'll get to this in another slide here too. But a diagram like this helps to understand this concept of half-life. From a measure of the original, from a knowledge of the original amount of an isotope in an object, if you know how long it takes for half of that isotope to decay, then you can measure the amount of that isotope at any particular time and estimate absolute age. Here from 100% quantity of that original isotope, half of it will remain because half of it will have decayed over one half-life. It's an exponential decay process, so that only 1/4 of that isotope will remain after 2 half-lives, and 1/8 after 3, and a 16th, and so forth, in a decay, in an exponential decay. Getting more specific here are a couple of isotopes. For example, uranium-238, potassium-40, carbon-14. These are unstable isotopes that decay to another form. Potassium to argon, uranium to lead, carbon-14 to other forms of carbon. And they have very different half-lives. The half-life of uranium-238 is 4.5 billion years. 1.3 billion for potassium, and just 5,730, to be exact, for carbon. Because of those major differences in half-life, these different isotopes are good for dating objects of different antiquity. Uranium and potassium are good for dating things that are really, really old. Potassium can date the oldest rocks on Earth. Whereas carbon is only good for dating things that are relatively young, like back to 100,000 years. You can't date objects more than 100,000 years old with carbon-14 because it decays too fast. There's just not enough of it left to measure for a fossil that's older than that. So any non-avian dinosaur is ineligible for carbon-14 dating because it's just too old. There won't be enough carbon-14 in it. Organisms pick up carbon-14 from the atmosphere where it's produced naturally. And we know how much there is in the atmosphere at any particular time. So we know that starting point. You have carbon-14 in your bodies. When you die, that carbon-14 will start to radioactively decay, turn into another form of carbon. So if we test your body some thousands of years from now, you will have sequentially less carbon-14 in your body, according to this process. So if we measure you and find that you only have 1/4 of your original complement of carbon-14 in your body, we know that you've undergone 2 half-lives of carbon-14, since there's only a quarter left. You're half and half again. You've undergone 2 half-lives of change. You must be 5,730 times 2 years old. And it's pretty darn accurate and precise. And even potassium-40 is good to within a very relatively narrow range. Some hundreds of thousands of years, maybe even less, plus or minus, in some of the estimates for the human fossil record. A couple million years, plus or minus, on some of the estimates for uranium. There's excellent precision here. Yeah. It's an assumption based, so the carbon-14, and I don't know too much about this, but it's formed through solar radiation hitting nitrogen, I believe, and forming these nuclides in the upper atmosphere. And it's a consistent and stable process such that the amount of carbon-14 breathed in by organisms, taken in by organisms, is apparently stable through time. And that's about all I can tell you right now, but we could look it up and research it a little better. It's important with volcanoes for these other elements because of volcanic eruptions essentially establish these baselines for many of the heavy elements. These heavy elements were produced not in the upper atmosphere like carbon-14. These nuclides were produced in supernovae, in stars, in ancient stars. And then making their way into the Earth can spew out in volcanoes at their original composition, at their original levels. Having spewed in a volcano, they then undergo the radioactive decay process. So oftentimes, unless you have volcanic ash layers, you don't have the opportunity to use absolute dating for the fossil record. That's true in Africa for the human record. You're stuck if you don't have good volcanic ash. If you have volcanic ash at one level, and then a fossil, and then another volcanic ash layer. So you had a volcanic eruption, something died and was fossilized, and then another volcanic eruption. See what you can do? You can't date the fossil. You can't date that bone directly because it's ineligible for something like carbon-14, which is present in the bones. You can't use potassium dating on the bones because there isn't the sufficient quantities of potassium in an organic being. But you date the ash below and you date the ash above, and you bracket the age of the fossil. So you know it must be younger than this and older than this. And depending how tightly that is, how tightly bracketed that organism is by the volcanic eruptions, you're on the right track. So with that, you now had a way to absolutely date this great geological record. These names, some of these names predate Darwin, but they didn't know how old these times were. They just knew that this one's older than this one. And they could still name it, and name it they did. But only after radiometric applications could absolute numbers be hung on this scale. And so in this great scale, no, you don't have to memorize it all, but certainly not. But it is good to memorize, especially if you're going to take more science courses at university. You don't need to relearn it every time. Just learn the basics now. And it's just the major names that I'll highlight on the slides and provide you in the terms list that will be the ones you need to know. And the dates that I want you to know are on this slide and the next slide. So remember, we have eons, eras, periods, and epochs, right? Well, we also have major extinction events that I'll focus on next time, and they form the boundaries between these major eras. But let's go through some of these landmarks. Another helpful way to look at it is in the form of a circular clock, using an analogy like this, because we're trying to get a handle on this deep time and finding some access to wrestle with this scale of 4.6 billion years, or 4.5, or plus or minus a little bit, about for the origin of the Earth. If that happens at midnight, by 3 p.m., you have your first fossils in the form of these stromatolites that we'll look at, these fascinating fossil preserved systems. The appearance of an oxygenated atmosphere, hugely important in the metabolic evolution of life on Earth at 2.7 or 3 billion years ago. I have 2.7 on here. Some recent evidence suggests maybe a little earlier. Doesn't matter. We're not quarreling over those differences. This is subject to revision, subject to constant revision, but we're narrowing down on some of these times. The origin of eukaryotes from prokaryotic ancestors. The origin of multicellularity, no date given, but just the fact that there were multicellular eukaryotes, all single-celled existence up until about 8 p.m., right? Animals explode around here, and creatures are getting large and diverse not until, 10, 15 or something, p.m. And humans not appearing until close to midnight. So, if you think about the last few hundred years, we're talking about just this narrow little slice of experience right there, right? So that can help, thinking about it as a clock, in terms of getting a sense of scale, sense of time scale. These guys were foundational in performing laboratory experiments to help illustrate the possibility of life having evolved on Earth through physical means. It was questioned how you could get the spark of life on Earth through strictly physical mechanical means. Darwin had a solution with reference to a warm little pond somewhere with the input of lightning could have triggered the early steps in the formation of the most basic molecules that would allow for a complex life. And that's pretty much what Miller and Urey were trying to replicate. Miller, the graduate student, Urey, the advisor. Miller says, I must try this in the lab, and Urey says, you're crazy. You've only got a short amount of time to complete a PhD. You're not doing it. He says, I'm doing it. And the story goes, the advisor said, okay, you've got 2 years, and if you don't produce something in the next 2 years, then forget it. Deal? And he said, deal. And he did produce in the laboratory through a really simple contraption that's been repeated and even elaborated on and found to hold a ton of information in subsequent studies. Just using water and an energetic input in the form of the heating of the water and turning it into a vapor and mixing it with some of the chemicals that are thought to have been present on the early Earth in the atmosphere, like methane and ammonia, and subjecting that to a spark to simulate the lightning with an electrode, condensing that water out and analyzing the result, finding compounds that could serve as the basic building blocks for amino acids. So just through a physical means, the synthesis of the basic materials that are known to exist in organisms on Earth. And for many people, this was really enlightening. This was a verification of the possibility of the basic building blocks forming through physical means. From there, if these little basic building blocks, these molecules, have some replicating ability, and they differ one from the next, you have the stage set for a natural selection process and microevolution. Archaeans were introduced in the last section, right, as one of the types of prokaryotes on Earth. In California, if you fly into California from the south, you can see these colorful ponds in the South Bay. Some of this color is given by the archaeans growing in the ponds, also algae. Or if you drive up north to a place like Lassen, I was there with you guys. Remember our hike to Lassen here? We were on a hike together when this picture was taken. As you may recall, you were little. But there are these thermal pools up there that include archaeans growing in the thermal waters. These are sometimes called extremophiles, right? Because they inhabit some of the most extreme environments on Earth. Very hot environments, very salty environments, deep water environments, the archaeans. And probably were an example of some of the earliest organisms on Earth with the bacteria. the other type of prokaryote. Stromatolites I referred to as some of the earliest fossils ever found. They are 3.5, 3.9 billion, somewhere in there. You find these fossils that have these pillow-like structures. And these things live today too. There are some shallow water warm lagoons today where stromatolites still grow. A fossil stromatolite like this has this complex layering where the organisms, the prokaryotes, the bacteria, have laid down these layers of material as they grew upward toward the sun, photosynthesizing, laying down sediment, and forming these complex bands that were fossilized. These are communities of prokaryotes that are constructing these stromatolites. These are biofilms. You basically have a few seed prokaryotes that can cling to a surface and replicate to form this community with this sort of slimy coat that generates a more complex structure with cavities through which the fluid may flow and the nutrients be captured. And this community is signaling within itself maybe multiple species of prokaryotes eating as it goes, as the fluid washes over it. Maybe it's photosynthesizing. And it builds up in this direction as it grows. That's a biofilm, technically. And guess what? The plaque on your teeth, maybe you're in pre-dental, want to go to dental school? Well, study dental plaque as what it is, a biofilm of a prokaryotic community growing on the surface of your teeth. Because if you take that approach, you're taking a legitimate ecological approach to this ecosystem that is your mouth. And that can be, the dentists are becoming wise to this, and some very excited about it. It's related to the microbiome concept that's become so popular in an analysis of the digestive tract. This is another form of microbiome, this biofilm in the mouth. Cyanobacteria, quote-unquote blue-green algae, right? Used to be a member of the broad group known as algae, but we recognize it as a good prokaryote, a good bacteria now. Blue-green algae, or cyanobacteria, blooms like crazy in appropriate conditions today, particularly in waters that are heavily fertilized, polluted, and warm. You can get these great blooms. Well, they bloomed like crazy on the early Earth, too. They bloomed to such an extent that they created a revolution in our atmosphere by photosynthesizing and producing oxygen. They released oxygen into the oceans. The oxygen in the oceans reacted with the iron in the oceans because the oceans were iron-rich. What does iron do when it interacts with oxygen? It rusts, right? It creates so the oceans essentially rusted through the production of all this oxygen, and you can see it in the fossil record. You see these bands of iron oxide that are laid down in sediments from this time period of 2.7 billion years ago as a result of the activity of cyanobacteria producing so much oxygen. Having filled the oceans with oxygen and knocked out so much of the iron, where was the oxygen left to go after the ocean became supersaturated with oxygen? It started to bubble out of the oceans into the atmosphere and began to change the global atmosphere, oxygenate the atmosphere. This was poisonous to most of the organisms that were already present because they were anaerobic. They couldn't tolerate the oxygen. So life flipped and came to rely on the oxygen in a new metabolic strategy and took advantage of this toxin. And sometime thereafter, we had the origin of the eukaryotes, many of which took advantage of oxygenic respiration. The eukaryotes, you were introduced to the story in Bio 1A if you took that, and probably in the first section briefly as well. The hypothesis that eukaryotic cells, originated from a prokaryotic ancestor invading another type of cell. There's strong evidence for that in the form of organelles such as mitochondria that have their own DNA, circular strands of DNA that resemble the DNA of prokaryotes that replicate independently of the nucleus in a eukaryotic cell. These are ancient prokaryotic invaders that became hosted by this cell and now form fundamental cellular components of the eukaryotic cell. It's an amazing story that was absurd 20 years ago and controversial until absurd 30 years ago, 40 years ago, controversial until 10 years ago, and now pretty standard, the endosymbiotic hypothesis. And there were more layers that occurred on top of that with the photosynthetic prokaryotes like cyanobacteria having invaded to form chloroplasts and the plastids in plant cells, right? I'll look back and see how much you've been introduced to in the previous section in order to see if I need to explain more on that. But we end up with a tree of life with these three great domains: bacteria, archaea, and the eukarya. The eukaryotic line having been contributed to by proteobacteria in the form of the mitochondrial organelles and cyanobacteria having invaded or been taken up in the origin of chloroplasts in photosynthetic eukaryotes. Photosynthetic eukaryotes tend to have both mitochondria and chloroplasts. We animals, well, we have the mitochondria, but not the chloroplasts, right? So in that sense, every cell in your body is involving a community process. It's a radical flip. And guess what? The countries that used to be most comfortable with this were communist countries. The communist countries, Russians originally devised the first endosymbiotic theories 100 years ago. They were entirely comfortable with the notion that even the cells in your body might be small communities. And it took the rest of the world a lot longer to come to terms with that. And so there's a political dimension in our thinking when we approach these scientific problems. It's for real. We're influenced by those types of bias. So none of this stuff is critical. Let me just tell you a few stories here to finish. None of this is detailed information that's really important to memorize or anything. But we need to grow our creatures to larger forms in order to pave the way toward the story of vertebrate evolution and human evolution as a narrative here. Multicellularity develops through a hypothesis maybe like this, whereby an anus forms and a mouth forms on organisms that are increasingly complex with multiple cells in the body. Organisms become diverse in form. This might not look like much, but that's pretty complex for 575 million years ago. This flat-paneled creature, Dickinsonia, with a head on one end and a posterior on the other end, at least a front and a back like that, with a trajectory through its watery system, to and fro, with this complex patterning of isomers. But the real explosion occurs right after, in the early Cambrian, around 542 million years ago. There seems to have been a legitimate revolution in forms, this short period of time where there was just this burst of activity. Quickly, in this span of time, all of the living phyla on Earth, and many more besides, were produced in some tens of millions of years maybe. Exceptionally preserved fossils from the Burgess Shale, a famous fossil outcrop, helps to elucidate that time. You can read a book like Wonderful Life by Steve Gould if you want to read about the subject. Fossils like these illustrate the diversity of the multicellular creatures at that time. Some of them are relatively easy to link to existing animals and other organisms, but some of them are also quite tricky, like this thing that has fooled people over the years, aptly named hallucigenia, because you weren't sure if it protected itself with these, or if you flip it upside down, if it walked on these, you don't even know which way's up. There's nothing in the world today to compare a creature like that to. So the debate has been about how it moved, and did it stand on those, or did it use those to protect itself? I think there's resolution to that, but I haven't entirely followed it. And then just finally, let's think about one of the transformations that happens here in the Cambrian. You've got a watery world of activity. most of the major dynamics of life are still happening in the oceans and waterways, right? And you have these waterways, and they have a floor. They have a sedimentary floor that's accumulated through a rain of material, of dying bodies and decaying materials to create this sediment on the ocean bottom. You have a terrestrial environment that's been invaded by prokaryotes and fungi and other stuff, but so much of the dynamics is happening in the oceans still. Organisms are starting to move seriously up into the water column, off of the surface of the floor of the ocean itself, and down into the floor of the ocean. It might not seem like much, but the beginning of the burrowing into the ocean sediments opens up an entirely new universe of activity and starts to circulate nutrients that have been piling up through all the decay of the organisms above for so many millions of years. And so burrowing, the formation of burrows, was an absolute revolution in the ecology of the planet. Trace fossils like these illustrate the burrowing of wormy creatures, some of which live today, these priapulids, that illustrate this revolution in ecology that triggered, yet again, more evolutionary dynamics. So the changing in the physical environment can stimulate these changes in the biological universe, and vice versa. It's a feedback system. All right, any questions before we say goodbye for the weekend? Have a great weekend.